Thanks for inviting me to this conference. It's my first conference, so thanks to the organizers for letting me present our research at the University of Wisconsin-Madison. A little bit about myself. I've only been in the cannabis space for maybe 6 years. This conference has been great. Yesterday I learned a lot of stuff. I've been in the biotech space for all my career, so about 35 years. Most of that in private industry for 30 years, and then I kind of came back to University of Wisconsin where I graduated from and directed a facility that I'll show in a few slides that does plant engineering and editing of lots of different species. And today I'm going to talk about the work we're doing in cannabis, and like Texas, I'm from Wisconsin, so we're dealing with a type 3 version, so I'll be interchangeably using hemp and cannabis during the talk. Okay, so a little bit about our team at the Wisconsin Crop Innovation Center. We're located off campus, and we're a facility that has about 50 people working at it. We're kind of almost like a small business. We have our own production, discovery, plant analysis teams, administration facilities, so it's almost a unit outside campus that does this work. The core hemp team, myself, Sean Kepler is the faculty director of the site. Shelby Ellison is an assistant professor working on campus, and she does a lot of the cannabis breeding and feral hemp work on campus. And then my team, Cindy, Lucas, and Jane, they're all working in the lab doing the transformation and gene editing. This is a bird's eye view of the center itself. It used to be an old Monsanto facility. So Monsanto donated this facility about 7 years ago to the university. I directed the facility for about 5 years, and then I stepped back and came back to work on hemp 2 days a week just because we had a grant for hemp, and I thought it was just a fascinating plant. It's really the first time I was introduced to it. The facility does transformation of many different species, corn, soybean, different species for both public and private industries, so they can pay a fee for service. We'll put your favorite gene or knock out your favorite gene in any of these species and hand you back T1 seed, and it's no strings attached. It's yours to do whatever you want to. We also do collaborations and research agreements as well if they're longer term. We were the first, I think, in the world to stably engineer heritable cannabis, and that was in 2018. We filed a patent for our procedure, and it was granted already. And like I said, we have a dedicated team of those folks working on cannabis research. Why hemp? You've probably seen this slide a million times, but some people call this a $1 trillion plant, but not only for medical uses, but industrial hemp, there's all kinds of uses for industrial hemp as well. Farm Bill legalized hemp in 2018, so that gave us freedom to really take a look at it because there's really very little research done in the last 80 years on it. And then of course, the rec use and the medicinal uses even increase the value of that. So I think really this is a disruptive time for cannabis, the cannabis industry. I put this slide up just to show that kind of the history of putting genes into plants. First functional genes in plants was back in '84. I was working with a group on this as well as some Monsanto employees and others in universities. That's just an old southern blot showing T1 inheritance of a gene in a plant for the first time. Then in '96, was kind of a pivotal year as well, where Roundup Ready was kind of the first, I'll call it GMO plant that was widely commercialized. There was, of course, the Flavor Saver tomato before that, but it was kind of a failure. But Roundup Ready ushered in this commercialization of GMO crops, brought in lots of no-till farming, glyphosate-resistant plants. Our group back in before '96 worked on what you see up there is the gene gun. So we invented that at a private company that I worked in. It was an electronic device that was used actually to make that Roundup Ready event before Monsanto purchased that company. And then in 2018, this is just a picture of our first transgenic event. That's a T1 picture showing a red fluorescent protein. This was just a proof of concept to show that we could do it. Progeny and the red plants in that picture are expressing a red fluorescent protein, whereas the green ones don't have it. So a little bit about genetic transformation in cannabis. Lots of labs have published and can do transformation in tissue culture, and they can get actual transformation to happen. But the key thing is most of them aren't able to make it heritable or stable. So they get it in callus formation, in roots, different parts of the plants, but they're not able to get a stable transformed event. It's very recalcitrant to tissue culture, and when I say tissue culture, I mean embryogenic tissue culture. So if you have a callus form of the hemp plant, very hard to actually get a plant to grow out of that callus to differentiate into the plant. So one of the things we've done since the 80s at this facility is we focus on meristem explants. So we don't deal with callus or organogenesis. We take a mature seed, we imbibe that seed overnight, and then we isolate the meristems with the cotyledons, or with the primary leaves on them. So this is a picture, let's see the laser. That's a picture of what our starting material is. So that's your root radicle, your primary leaves there, and then there's the meristematic region there that we're targeting for transformation. We've been able to use both Agrobacterium transformation and the gun transformation for this. We choose Agrobacterium just because it's much more efficient and straightforward. Oops, sorry. If you don't know anything about Agrobacterium transformation, if you go for a walk in the woods or in the town, you see plants with crown galls. So it's these massive formations on these plants. That's basically a soil bacteria that's infecting these plants. Naturally, it infects these plants, and it has some oncogenic genes that form this crown gall. So it's almost a cancerous growth on these trees. And scientists over the years have hijacked this bacteria. They take out the genes that form that crown gall, the cancerous genes, and then you can put in whatever gene you want to. So whether it's a yield gene, an insect-resistant gene, hop latent mRNA potentially, and that plant will infect the cannabis plant and deliver its genes to the plant, and it'll express that trait. So we start out, this is an Agrobacterium plasmid. We start out with a plasmid that's located inside the bacteria, and this is what the bacteria is going to deliver to the plants. And this is where you put your whatever gene you want the plants to express. And I'm just showing you the the proof of concept vector that we used. Whoops. The proof of concept vector we used, and I mentioned it had a red fluorescent protein in it. That would be this TD tomato gene, it's called. It also has a Gus gene in it, which is a gene that if you give it a substrate, it'll show a color of blue if the gene is present in the plant. So these are just visual markers that allow us to track, did the plant get transformed or edited. So those embryos I showed before, those go through an antibiotic selection. We use spectinomycin as the antibiotic, and they get put on media plates for about 3 or 4 weeks. You can see some bleaching here occurring. So if the plant doesn't have the spectinomycin gene, that spectinomycin will start to bleach the plant and eventually kill the plant. Anything that's greening shows that plant may have the spec gene in it, and it's probably transformed. And along with the spec gene comes your gene of interest, TD tomato, Gus, a yield gene, or whatever. Usually this process takes about 6 to 8 months, going from transformation, or co-culture of the Agrobacterium, to getting T1 seeds out the door. This is our first T0 transgenic event. I mentioned it had this blue Gus gene, and we did some staining, and you can see this expression of blue within the germline tissue of this plant. So we did this as a pretest to see if it was transformed, and indeed it had blue in the germline tissue, so we were pretty positive that it was a germline transformation. Excuse me. So a few statistics on our transformation procedure. Our frequencies are in the single digits. So meaning if you take 100 of those embryo explants, we may see 1 to 5 plants come out that are transformed. Normally for soybean or corn, we see that in the 20 to 30% range. So we've got work to do to increase that frequency, but it's very repeatable. We've had success with multiple cannabis genotypes. And again, we're from Wisconsin, so I'm using CBD varieties. I don't think this particular procedure is going to be genotype dependent. I think we can do marijuana, fiber, hemp, whatever it may be. We've made over 200 independent events for private companies and internally, so it's, again, very repeatable with whatever we're trying. We've also done gene edits, and that's what I'm going to get to in the next few slides. And then, since we're starting out with a embryo explant, we're not starting out with a single cell, we do get chimeric events, anywhere between 10 and 20%. And a chimeric event is just an event that's not fully transformed in every cell, but it is a germline transformation normally. So if you take that plant out to T1 status, it may not segregate 3 to 1, but you'll be able to find your true germline transformation and then take it forward, and it won't be chimeric. That's, again, a picture of our T1 progeny. Some other examples of our T1, so that was the TD tomato. We put a gene called Ruby in. Ruby is it's a set of 3 genes that make beets red. So if you eat beets, that's the gene. So we put that into hemp, and we get a plant that's totally red compared to the null. And this is an overexpression of that gene. Then we've made knockouts, we've made overexpressors, and we've made some other plants for private companies where they're using evolved CBD synthase or THC synthases to see if they can change ratios in the cannabis plant. I probably didn't need this slide, but normally we're dealing with a variety we call Badger. It's got some cherry wine in it, it's got some Chardonnay, it's got some Electra in it. That's kind of the background. They are dioecious plants. So we have to feminize them or masculinize them to make feminized seed in order to get the gene to be homozygous in the next generation. So I mentioned our frequencies are in the low single digits, so we're doing a lot to try to increase our transformation frequencies at the center. We're trying different selection agents. So I mentioned we use spectinomycin right now. We're using different agents. We're using different Agrobacterium strains. We're trying liquid culture rather than a solid media. Probably the biggest thing to try for us right now, as well as a lot of other species that people are trying, are these morphogenic genes. So these genes elicit embryogenic responses in plants that are recalcitrant to transformation. So things like baby boom, wuschel, LEC2, these are all genes that a lot of the big ag companies have looked at and are getting really good results with, especially with dicots, or especially with monocots, and now recently with dicots as well. So we're going to be looking at this as well. Yesterday they talked a lot about LED lights in culture, in cultivation. So we're looking at LED lights also for tissue culture, so pre-cultivation, and whether we can elicit better growth or different responses directly in tissue culture. Media and hormone components are always played with for probably the last 40 years, so we always look at those. And then other target tissues other than the immature embryo. So targeting, so after our proof of concept was done, we thought about targeting different enzymes or genes in the cannabis plant, and some of the obvious ones were, the cannabinoid pathway genes, the terpene pathway genes, to look and see if we could actually go and knock some of these out, to see if we knock these out in the plant, we got similar results that they have done in yeast or bacteria, where they've knocked these genes out. So one of the questions, and it was a controversial question for a while, but why do CBDA type 3 plants make THC? And Medicinal Genomics did a lot of work on this. The hemp plants that we use, the type 3, they don't have an active THC synthase. So the CBD synthase makes actually a little bit of the THC. So when you get your 0.3 or 0.4 whatever percent THC you're getting, it's the active CBD synthase that's actually making that. It's a promiscuous enzyme. It's very homologous to THC synthase. So that's what we targeted in order to try to knock out CBD. And the reason we went after this was back in 18 when the farmers, or 17 when the farmers were growing their first fields of hemp in Wisconsin. About 25% of the fields went hot. They had to destroy their plants. They were saying, we need a THC-free line in the states that get hit for that. There are some CBGA lines, which is the grandmother cannabinoid of that. There are a few lines out there in 18, but not too many. The genetics of those lines are a little bit questionable based on some data that was done at the University of Wisconsin. There was also, not in 18, but recently GTR made a low THC line that was high in CBG as well. So we went after that CBDA synthase to knock out, and we used this CRISPR-Cas system, which they use routinely in other crops. Basically what you end up doing is you design a guide RNA that targets whatever sequence you want to knock out. That guide RNA sits down on the sequence, Cas9 comes in and makes a cut at that guide RNA, and then the plant itself, once it's cut, it religates itself, just a non-homologous end-joining religation event. And hopefully, when that religation happens, you've either dropped out a base or added a base. Somehow you've disrupted that gene. And that's how gene editing works. So we targeted that, the CBDA synthase in our Badger line. I'm just showing a couple examples of what we got out of that. The first plant we sequenced. We had actually 2 guide RNAs that we sat down to try to increase our chances of it working. The first guide RNA did cut, and we added a single nucleotide to this sequence, which would knock it out and make it non-functional. But we also had an insertion also at the other guide site of about 177 bases. And this came from the plasmid that was introduced into the plant. So that was the first event. The second event, the 2 guide RNAs cut perfectly at where they were supposed to cut. And in between those 2 guide RNAs, the whole sequence just dropped out. So we lost that whole sequence. The sequence came back together. And again, we have a non-functional CBDA synthase. And if we're successful with this, we shouldn't make any CBDA in the plant. And since the CBDA is responsible for the THCA, we should not have any THC in the plant as well. So then we looked at HPLC results. This was done on, I think, 5.5 week flower. And you can take a look at, take a look at the wild type plants here. You can see the CBDA is high in these plants, and then there's a little bit of THC as well, which that's what this plant would normally make. The heterozygous edit, and I failed to mention, but when you make an edit, sometimes you'll only edit one allele in the plant, not both alleles. So you need to edit both alleles in order, for that gene to be knocked out. So we went through a round of selfing of that plant in order to get a homozygous lines for this plant. So we had heterozygous lines which just had one allele knocked out, homozygous lines which had both alleles knocked out. And you can see the heterozygous look very similar to wild type results, and the homozygous edit completely drops out the CBD and the THC, and then the precursor, which is CBG, elevates levels because there's nothing pulling on it anymore. So we have a high CBG line. Again, these were only at 5.5 weeks taken. Normally this Badger line is somewhere in the teens for CBD, so we expect when we test this at later stages, we should be in the teens for CBG. This line, also we applied to USDA for non-regulated status and got that approved a couple months ago, so you can freely, this is an edited plant, you can freely plant it out in the fields without regulation. And we call it Badger G, just for, we're the Badger state. So next step for this Badger G line, we want to fully genome sequence it. We're bulking up feminized seed right now so that we can get larger quantities for people that may be interested in it. And again, this is just a high CBG line, so there are other CBG lines out there, we realize. But back in 18, this is what we were after. There's companies, Hypercan is a company that's interested in this line. They want to have a blank slate to start with. So a high CBG line would be optimal for them to put in their engineered CBDA synthase genes so they can play around with ratios of THC to CBD. Penn State's interested in this for animal feed, so since it doesn't have any THC whatsoever, it gets past some of the regulation that some states have for animal feed. And then we're crossing this line, since our line is kind of an unknown, this Badger line's unknown, it's a cherry wine-based background. We're crossing this line with better known CBD varieties like ACDC and some others to get into hopefully a more well-known cultivar to people. And then the other thing we're going to do with this line is take a look, since we have a lot of CBG available. CBC is another minor cannabinoid that we've heard a lot of people are interested in having a very high CBC line. So we think by using this line and introducing either mutations to the CBC or overexpressing the CBC synthase in this line, we should be able to make a 10% plus CBC line. So other ongoing research is that we're doing besides cannabinoids, increasing flower and trichome production to try to just increase overall cannabinoid levels. Biomass and yield, which is with any crop valuable. Flavonoids, we're looking at the flavonoid pathway to see if we can increase maybe canflavin A, canflavin B that can be used in medicinal studies. Fiber properties, so industrial hemp is up and coming. So a lot of people are asking me, what can we do for fiber in hemp? Can we make more uniform fibers, longer fibers, reduce certain lignins in hemp, things like that. So we'll look at fibers. And then seed and nutrition. Can we do anything with seed? So again, I find this very similar to what they were looking at in the 80s back for row crops. Think about any trait you want to put into hemp. If you don't think you can do it by breeding or molecular breeding, or it's going to take too long, but this has potential. Disease. So I got sandwiched in between two hop latent talks, and that's all I hear. I hear a lot at this conference. So hop latent is another target that we're doing. It's been shown to, the RNAi technology has been shown in other species to work for other viruses and viroids, so we're going to try to attack that, as well as powdery mildew. So there are, again, other species like wheat and others that people have been successful at getting powdery mildew resistance in, so we're going to try to do that in hemp as well. Terpene profiles are another area that people are interested in. So increasing levels of various terpenes that are desirable, increasing trichome density, and maybe knocking out certain terpenes that people don't want to have, so you can customize your smells or flavors or whatnot with different terpenes. Herbicide resistance, it's always out there. I have larger companies asking that are looking at hemp for paper. They claim weed is a problem in their large fields, so they ask about that. And then just a few slides on collaborations that we have, and these were just press releases, so that's why I put them up. ZCal was a customer that came out and put their photoseed trait in hemp, and they got an increase in biomass oil. And then Hypercan, this should say actually 2023, but Hypercan put their evolved CBD synthases, which are supposedly very active and produce more CBD in the plant into hemp as well, and they saw an increase in cannabinoid content. Again, Hypercan uses a lot of X-ray crystallography. They know this enzyme inside and out. They know what mutations can change the CBDA synthase to make different ratios of CBD to THC, so they can customize that. And I think the last one on Hypercan is, this was just their data from their press release. So showing an increase in leaves and immature flowers in different cannabinoids, mainly the CBD and the THC. And I'm going through these pretty fast because I'm running out of time. And then we're also looking again at the CBC, because there seems to be a lot of interest in CBC. So can we make a CBC line that is 10% plus, 15% plus for the community? And this was kind of hot off the presses. I thought I'd throw in there. We got this result last week. It's very early. It's just on young leaves, but we targeted also the OAC enzyme, which is olivotolic acid cyclase. That's olivotolic acid and GPP are the two precursors to CBG. So if you knock out one of them, the plant ideally shouldn't make any cannabinoids at all, and that's what we're seeing in this early data for leaf data. So our control line makes some CBG, CBGA, and our OAC knockout basically doesn't make, we're still looking at these bars here. They're kind of right at the limit of detection for our HPLC, so we don't know if they're real or not, but it's basically dropped out the CBG out of this plant. So I came to this conference, trying to open minds and trying to think about, what traits are out there that people are very interested in, looking for collaborations, partners, anybody interested in, putting their favorite trait or knocking out their trait in hemp can come talk to me after the talk. And that's my last slide. Questions?
Do you happen to have any pictures with you of the early transfer, early regeneration process? Once you transformed these meristems, maybe you have some more.
Other than the ones I've shown.
Yeah. That's how it looks like when they come back from, regenerate from that little virus.
I'm sure I could dig up some for you. We can, yep, yeah.
Hear me? All right. On the screen.
Great talk. Excellent work. How large of an edit or gene do you think you can fit in that vector, and how do you handle multi-exon genes?
Yeah.
Genes there. But obviously, the cannabinoids are single exon genes.
Yep. Yeah, so there's a couple different answers, I guess. If you want to knock out multiple genes, just design your guide RNAs to knock out whatever gene you want. Normally, binary plasmids in agro, normally you can make probably up to a 30 kb vector and have it stable. We've been playing around and have had success with a new, I showed it up there on one of the slides, it's a vector called gantry. So it doesn't use a binary vector. We put the genes right in the backbone of the agro and deliver it through the backbone of the agro, and we're able to make easily over 40 kb size plasmids to put in. So if you wanted to do overexpress, say, a multi-cistronic pathway or whatever, you could do it using that gantry vector. Yep.
Mm-hmm. Is the efficiency strongly reduced by the large size of these plasmids, or can you still get decent numbers?
Yeah, efficiency normally I would say goes down just in general in all species. Once you hit say 15 or 20 kb, delivering that tDNA size, that's where it just starts dropping off. You can still deliver over a 30 kb thing, but your efficiency of delivering that whole thing is going to go down. The nice part of the system is, you put your selectable marker on the left border, right? So you're sure that the whole thing is going to go in and read through the whole thing. Yep.
And you mentioned that you're working with different auxotrophic strains of Agrobacterium. Have you seen any significant differences?
Yeah, it's kind of early work. We're doing some LBA 4404 Thymine strains or thymidine minus strains. The reason we're doing that is just agro overgrowth. We're trying to control the overgrowth in the agar. We use timetin, we use carb, we use ceph, but every once in a while we'll get this cropping of agro overgrowth that tends to lower efficiencies. The thymidine minus, I think it's still out whether they're as efficient as other vectors. I've seen certain cases where certain auxotrophic vectors don't perform as well as in delivering tDNAs.
So you're working with LBA as opposed to like GB3101 or?
So the work we're doing here, and I mentioned LBA because that's kind of that's the auxotroph we have right now.
Okay.
The work we're doing in cannabis right now, we've done GV3101 is the one vector we've done, but we mainly use a strain called 18R12V. So it's a K599 based Ori-RI type vector that was disarmed. So, and we use that mainly because it gives us a little bit better quality of an insert, so we get potentially fewer copies going in with that particular thing because it uses an Ori-RI origin. Okay, thanks.